In this week's lab, you're going to be working with and understanding fermentation. And fermentation is simply regenerating oxidized form of NAD, so NAD plus, for glycolysis in the absence of oxygen. So let's take a look here. So we have our black box of glycolysis, right? That's 10 enzyme catalyzed reactions that convert glucose into pyruvate. And then in addition, we get couple NADHs, right? And we get some ATP from substrate level phosphorylation. Now, when there is oxygen around, we can recycle these reactants, right? In order for glycolysis to occur, we need glucose, obviously. We need a supply of ADP and inorganic phosphate, and we need our oxidized electron carrier. We need NAD+. If we run out of any of these, then glycolysis will shut down. Now, in the presence of oxygen, that's not a problem because any of the NADH that gets made is going to be taken to the matrix. It'll dump off its electrons there in the matrix for the electron transport chain and thereby oxidizing itself back into NAD+. Now, to recycle ADP and inorganic phosphate, it doesn't matter whether there's oxygen around or no oxygen around. Anytime a cell uses ATP, it sort of recycles itself back to ADP and inorganic phosphate. And then in the presence of oxygen, pyruvate goes into the matrix, becomes acetyl-CoA, and then goes down into the citric acid cycle. Now here is the problem. Without oxygen, the citric acid cycle and the electron transport chain both stop. Because they are completely dependent on oxygen, which means that pyruvate's no longer useful, right? That's not going to go to the matrix anymore because the acetyl-CoA can't be used in the citric acid cycle, and NADH no longer has a place to drop its electrons off. So we lose this recycling here. So that means that if it weren't for some specific solution that we will get to in a second, that we would start to run out of NAD plus in the absence of oxygen, and glycolysis would eventually shut down. However, we have fermentation. And what fermentation does is it says, okay, if without oxygen, if we don't have any oxygen, pyruvate's just going to be sitting around there. Let's use pyruvate to oxidize NADH back into NAD+, so that we can continue glycolysis over and over and over again, which means even though we're not getting any ATP from the citric acid cycle and the electron transport chain, we're getting 2 ATP every time we break glucose down into 2-pyruvate. That NADH gets reoxidized by pyruvate, and we can continue this cycle over and over and over again, thereby making 2 ATP each time. So ethanol fermentation is one of these ways that we recycle NAD plus using pyruvate. So here's our glycolysis. Here we're making the 2 ATP. Here we're making pyruvate. And here we are making our NADH. Well, fermentation is converting that NADH back into NAD plus. How does it do that? First, the cell has a reaction in which pyruvate is decarboxylated. There's your 2CO2. Right, that's going to be important. That's going to be our assay that we're going to use for whether or not fermentation is occurring by measuring the amount of CO2 in today's lab. And when you decarboxylate pyruvate, you end up with acetaldehyde. And it's acetaldehyde that is going to directly oxidize NADH back into NAD+, thus allowing glycolysis to continue. When acetaldehyde steals those electrons and hydrogens from NADH, it becomes ethanol. So there's another way we could have used to measure whether or not fermentation is occurring. Okay? But it turns out it's easier to measure CO2 production because that's a gas. All right, now, the organism in which we are going to study fermentation is yeast. And yeast is a single-celled fungi. It is a facultative anaerobe, meaning that it could live in the presence or the absence of oxygen. Okay? And without oxygen, they undergo a type of fermentation that we just described, ethanol fermentation, okay? Thereby producing ethanol and CO2. So that's why we use yeast to make beer. There's the ethanol. That's why we use yeast to leaven bread from the CO2. Okay? So that yeast fermentation is important, not only for the life of the little yeast cells here, but also for some yummy stuff for you and I. Now, the assay that we are going to be using is called a Smith fermentation tube, and it looks like this because it allows us to measure the production of CO2 given off by yeast fermentation. How does it work? Well, we take and we mix yeast solution with a specific test condition. And there's all sorts of conditions that you're going to be testing today. The effective pH on fermentation, what sorts of carbohydrates can yeast ferment, and so on and so forth. Well, you're going to mix that yeast with those specific conditions, and then you're going to pour that solution into this Smith fermentation tube. Then you tip it back so that all of this liquid goes all the way and fills this. Okay? And then every 10 minutes for an hour, okay, you'll come in and you'll see how many air bubbles, what is the volume of the air bubbles that accumulate up here. That is CO2 that's been given off from the yeast in solution. And we're going to measure that in milliliters. So for example, it looks like this is about 1.5 mils of CO2 that has accumulated in that Smith fermentation tube. Okay. Now, what are we going to be testing? There's 4 different categories that you're going to be looking at. And the first is, what carbohydrates can yeast use for glycolysis and fermentation? Okay. And so here, basically, there exists all sorts of enzymes within the biome that convert various carbohydrates, glycogen, starch, galactose, mannose, glucose, right, into intermediates of glycolysis. And you've got to get into an intermediate of glycolysis for you to be able to ferment, alcohol fermentation. So theoretically, right, organisms can ferment fructose, can ferment mannose, can ferment galactose. Right? They would have to have the enzymes necessary to convert them into intermediates of glycolysis. And so that's sort of what we're testing with yeast. So which carbohydrates are you going to test? Okay, you're going to mix a solution of yeast with each one of these carbohydrates. And then you're going to measure the amount of CO2 that is accumulated over the course of an hour. Now, in the meantime, I'd like you to go through and tell me what type of carbohydrate each one of these represents. Is it a monosaccharide, a disaccharide, or a polysaccharide? Next, you're going to want to know, okay, we found out which of the carbohydrates yeast can ferment. The next question is, what about the others? What about proteins? What about fats? What about nucleic acids? Can yeast ferment those? Well, again, theoretically, there are enzymes that can convert proteins to amino acids to intermediates of the citric acid cycle. Fats, the glycerol fats, can be converted into intermediates of glycolysis. So the question is, does yeast have those enzymes? The enzymes necessary to convert proteins and fats into intermediates of glycolysis, and therefore being able to then ferment them. So you're going to try, our control is glucose. We know that yeast can ferment glucose. But what about oil? What about albumin? What about DNA? These are 3 different types of macromolecules, and I want you to fill that out. We're going to do the same thing. Mix the yeast with each one of these different carbohydrates in 4 separate, or each one of these molecules in 4 separate experiments, and then measure the amount of CO2 that accumulates over the course of an hour. Next, we're going to look at, specifically, we're going to use a fuel source that we know yeast can ferment, that's glucose, and then we're going to see how metal salts might affect the yeast fermentation of glucose. Because it turns out that many enzymes use metal ions as cofactors that are necessary for them to catalyze reactions. In fact, 6 out of 10 enzymes in glycolysis require magnesium ions. And so if we take a look, this isn't an enzyme of glycolysis, it's DNA polymerase, but you can see very nicely here how it uses 2 magnesium ions to sort of stabilize the negative charges of the incoming nucleotide and orient that nucleotide in the correct way. And that's exactly what these metal ions do for enzymes. They're involved in electron transfer, orienting, and stabilizing the substrate, which means that if we add more magnesium ions to a solution in which this enzyme is dissolved, we might be able to get that enzyme to work a little bit better and act, and therefore the metal ions would act as activators, right? If they increase the fermentation rate over the control, right, over glucose with just water added instead of any of the metal salts. Or if we add other metal ions that can displace those magnesium ions and then stop the reaction, they might act as inhibitors. Okay, and so that's what you're going to do. You're going to take a yeast solution, you're going to add water, that's your control, no metal salt, right? Just yeast and glucose. And then you're going to take yeast and glucose and add sodium fluoride, copper sulfate, magnesium chloride. And you're going to measure how much CO2 is produced over the course of an hour. And finally, you're going to look at how pH might affect the ability for yeast to ferment glucose. So we're going to add glucose, a known fuel source. We know that yeast can use glucose to ferment. And then we're going to see, well, what's the effective pH on that? So this time you'll add your glucose, you'll add your yeast, and then you will pH the solution in these from 3 to 10 here. Pop that into the incubator and measure the amount of CO2 produced over the course of an hour for every 10 minutes. Now, this data that you're generating with this table, with this table, with this table, and with this table, you are going to make 4 different multi-series graphs. And there's going to be a document that will teach you how to do that. But I just want to point out here, I want to make sure that you label your x-axis, that you label your y-axis. This is the CO2 production. Make sure that you put the units here. So mils, I know I missed that there. I was a little lazy with this graph. And make sure that it's a scatter graph in which you don't connect them by lines. Instead, you add on top of that trend lines. But all of that is described to you in that Google document.